Hello, and welcome back to Scanning for Intelligence. What is a biology? Today we're going to talk about proteins, specifically amino acids. These are the constituent parts of a protein. So effectively, you can think of all the proteins in a cell as words on a page, and all of the letters that make up the word are these amino acids. So this word that is made can look like this guy, except typically these words are like hundreds of letters long. So like think James Joyce if he just kept it going. So proteins are made up of something called a peptide. This is a constituent part that follows like this. So you have a group like this and like this. So each of these are a repeating unit. So first of all, what does this mean? Like seriously, what does this mean? These are just a bunch of fancy lines and letters on it. And like a plus sign. It's weird, man. And what that actually means is this. So this structure right here, this up, down with some double lines, some weirdo stuff. So what each of these points represent on this is a carbon atom with its constituent hydrogens. Every letter like this is an oxygen. Sorry, O is an oxygen. N is a nitrogen. H is a hydrogen. In general, carbon bonds with 4 atoms, or it has 4 bonds. Hydrogen has 1, oxygen has 2, nitrogen has 3. If you have oxygen with only 1 bond, it's usually negatively charged. If nitrogen has 4 bonds, then it's positively charged. That's in general what happens. And so the positive and negative charges result in different characteristics of these proteins. And these different characteristics cause them to be hydrophobic or hydrophilic. Hydrophobic in general means it hates water. That's carbon and hydrogen. These atoms just don't have a distribution of charge like oxygen and nitrogen. When oxygen and nitrogen are present, usually they're hydrophilic, which means that they're able to interact with water, and water likes it, and so they get solvated by water. This leads to, in general, a protein structure like this, if you will, where the center of it is hydrophobic, but the outside is hydrophilic. We'll talk more about that next time. So first of all, we have this. So now that we know that each of these guys are carbons and hydrogens and oxygens, we can discuss what does this R mean? I don't know an R element in the periodic table. It's got these numbers also. So what this is just a general organic chemistry thing for a side chain, for something organic that's just kind of slapped on there. And that slap on is everything about proteins, essentially. So you have the peptide bond, which is this double bond here with the oxygen to carbon to the nitrogen with the hydrogen, and then these R groups attached are the functional groups. And in general, there are 3 types of functional groups: hydrophobic, charged, and polar. And then you also have miscellaneous category right here talked as weirdos, because nothing can perfectly fit in every single time, as we know. There are going to be those guys that in high school that are on the football team, but also AP students and just nerdier than all get out. And you have that just don't fit the straight, typical stereotypes. They make things interesting. But we'll stick first with the uninteresting guys, the hydrophobics. These guys are the guys that are buried in the center of the protein itself. So if you take it here, I draw a nitrogen, hydrogen, and an oxygen here. So if you just look at this guy, so then this R3 group is this guy. So right here, we just have a single line, which means we have a methyl at the end. What is methyl? Methyl is this. I think I might have forgotten to say, but 2 lines means double bond. So In general, that means it's a closer, tighter connection. Apologies about that. So alanine has this methyl, which is just CH3. And then, okay, cool. It's just a little funky guy in the end. Then you have valine, which has 2 methyls attached by methylene. Methylene is CH2. So that guy is pretty much the same as this guy, just a little bit bigger. And then you go out and you go to leucine and isoleucine. One thing I also forgot to state is that essentially this distance right here from carbon to carbon is 1.5 angstroms or 154 picometers. A picometer is 10 to the negative 12 meters, so extremely small. And trying to graph you around about how small, it's not really worth. And so it's just general best, just think in terms of angstroms, which is, so essentially this is 1.5, essentially any carbon to heavy atom is around the distance of 1.5 angstroms, and carbon-hydrogen bond is around 1 angstrom. There are definite deviations, and usually carbon-hydrogen, carbon-nitrogen, carbon-oxygen is slightly smaller, but having a number like that in your head, it just helps. So you have this guy, which just sticks out a little, 1.5 angstroms. Then you got valine, which sticks out just slightly further with a little bit more to it. Then you have leucine and isoleucine, which are pretty much the same thing as valine, just a little bit longer. These guys are just pretty much the same freaking thing. Huh. Now you have methionine. This guy has a little bit more interesting. There's an S right there. It's kind of just, like leucine, just got an S. What is S? Sulfur. If you remember the periodic table, sulfur is right underneath oxygen, but because it's below, it's less electronegative and has less polar properties, and it's also smack dab right between 2 carbons. Therefore, it doesn't have a lot of polar properties, thus it fits in the hydrophobic category, and so there's not a lot of interactions. These guys in general just don't interact with anything besides just kind of bumping into stuff. That's really their only purpose in life. Now we have these 3 guys, which are so hydrophobic, but they're a little more interesting. They got this ring, got these rings here. That means that they're aromatic, is the fancy person term. So you got this double bonded ring, this hexameric ring of 6 carbons. What is interesting to note is that I could have drawn these 3 bonds and just done here, here, here. Effectively, all of these carbons are double bonded with each other. It's this resonance structure. If you remember anything from basic chemistry, where essentially everything is just bonded with everything else. And so phenylalanine over here is just a basic hexameric ring attached to essentially an alanine. Then you have tyrosine, which is the same thing as phenylalanine, but with an hydroxyl group, OH. And that guy is in general polar, but because you have all this attachment attached to this, it doesn't have as strong of polar properties, so it's hydrophobic. And then you have tryptophan, which has 2 rings. You got a 5-member ring with nitrogen and hydrogen over here, and a 6-member ring over here. And so you have this, this, and this. So interestingly, these guys are able to insert into different cavities and in other proteins and able to do strong interactions. Additionally, when we go talk about amyloid plaques later, these guys actually can stack on top of each other like this, and the electrons all between them actually interact and have this steady connection, causing these amyloid plaques to form, which causes neurodegeneration in the patient, which is very unfortunate. And those bonds are very, very strong. So these guys are starting to be a little more interesting. Now we're going to get to the most interesting guys, the charged residues, specifically the positive charge. Plus Q means positive charge, just some physics notation. So arginine, histidine, lysine. Arginine has a very long chain with 3 nitrogens on the end. And these nitrogens are cause this positive charge, and this residue, this amino acid, is extremely important in a lot of different proteins, such that this guy sometimes even has the name of arginine finger, because it kind of just goes in and attaches to this molecule called ATP that is used as the source of energy in the cell. You know, mitochondria, powerhouse of the cell, makes ATP. ATP is the power of the cell. So then we go to histidine. This one's not as commonly used for reactions in catalysis, but histidine also has positive possibility of charge, which is shared by the nitrogens up there. So it's important. It's got an aromatic, so it can also have some sneaky little shoving into cavities for stronger binding to different proteins. And then we finally have lysine, which is very similar to arginine, except it's only got this amine group at the end. Amine is just essentially nitrogen bonded to hydrogens. That's just an amine group, and it's usually typically at the end of the chain. So arginine and lysine, we're going to be looking at these guys quite a lot later. Now we have negative charge. So these are different amino acids that have negative charge. So they have more electrons than protons. And we have two guys, aspartic acid and glutamic acid. Usually you might hear them called aspartate or glutamate. Yeah. And so these guys have two oxygens at the end. And the only difference between these two is they have a slightly longer carbon chain. These guys, they're interesting. And so they have negative charge at the end? Okay, cool. So these guys interact primarily with water because they are on the outside and they have the ability to interact with the polar nature of water, which, as we probably all remember, oxygen has a slightly negative charge, hydrogen has a slightly positive charge in water, which causes a dipole moment, indicating that you can have like a hydrogen here of water. We'll just do like this, H-O-H. So it's slightly positive, and these guys will interact with a hydrogen bond, although typically water is not stable enough to do that, typically. So now, polar residues. These guys are not charged, but they have this intrinsic property that they can still interact with water favorably. First of all, you have serine and threonine, which are pretty much the same thing, just get an extra methyl on threonine. And so, yeah. Because oxygen is highly negatively charged, typically, it will interact in a very similar manner to aspartic acid, and aspartate and glutamate. And then you have asparagine, and glutamine, which are pretty much the exact same thing as aspartate and glutamate, except instead of a negatively charged oxygen, you have a slightly positively charged amine group at the end with the nitrogens. So this nitrogen is slightly positively charged and will interact with that oxygen more favorably on the outside of this protein molecule. Now finally, we got the weirdos. The guys that just don't fit into a simple category, but we love them. Like Leibniz. He created the integral notation for calculus, but he was also a philosopher. With one of the most outstanding philosophies of everything is beautiful. We love him. We love that man. So, like Leibniz, we have weirdos. We have cysteine, which it looks a lot like a guy that we just saw, serine. So instead of oxygen, it's sulfur. Now, sulfur is still electrophilic, so why isn't it just polar? Now, here's the reason, is that cysteine actually, in our little fancy guy, it actually forms something called a disulfide bond, such that this hydrogen gets removed, and two of these guys get really close together, and they form the sulfurs actually bond with each other inside and help the structural formation of the protein itself. Very interesting stuff. It's super common in antibodies, such as those that are made by different companies for the treatment of coronavirus. Finally, we have glycine. Glycine is possibly the most flexible residue of them all. So this guy can be on like the ends of loops in proteins, such that if you have a region that needs to be extremely flexible, this guy can take that string of being flexible such that all the other interactions remain the same. It's very powerful, like alanine. So usually you'll see glycine and alanine at these points of high stress and curvature such that they can hold this stress. And finally we have proline. Proline, that doesn't look like anything else we just saw. You're lying to me. You're lying to me. It's not an R group. It can go backwards. And yeah, this is the weirdo. This is the weirdest weirdo of them all. But we still love it. We still love it. Oh, it's a P. So this, it goes back and it's pretty much the exact opposite of glycine. It's extremely, extremely rigid. It can't deform all those same ways as in high curvature. It helps actually keep the structure a lot more stable and less flexible. So it's got the exact opposite purpose as glycine. So as I just drew out here, this little one letter is called the FASTA code. It's this just simple way of noting what amino acid is what amino acid, so that you can just kind of look at a sequence of amino acids in a peptide or protein and just be able to say, okay, it's AVI, so it's alanine, valine, isoleucine. So that's in general everything that's all of the amino acids and a little bit more information about them. Typically, the protein on the outside is hydrophilic, which is formed by charged and polar residues, and the inside is primarily hydrophobic. And with that, we have all of the amino acids shown here. And I would like to just say thank you. It's been Mr. Johnny Hopkins, and I'll see you next time.